Okay, so today we're going to start talking about chapter 28, and this is going to be the reproductive system. We'll first talk about the general things about reproductive system, and then we'll focus on males and females and the differences there between them. And then chapter 29 will be on development and heredity. So we'll be basically looking at what happens if the egg cell from a female and a sperm from a male fused together. So we'll look at that next time that I'm talking to you. So let's go ahead and focus on this reproductive system. Reproductive system is a system that's responsible for making sure that the species continues, and it is. Sorry, I have a really loud jets flying over me. I don't know if you can hear that. So let's go ahead and look at some things about reproductive system. I want to pull up this table right here. Why is it not showing up? Okay, here we go. Okay, so the main organs of the reproductive system are gonads, and these are responsible for formation of gametes. Gametes are sex cells. And then we'll be also talking about a bunch of accessory reproductive structures, and these are going to be different, obviously, in males and females. So in this table, we are looking at some reproductive system homologs. And homologs basically mean that these structures originated from the same primordial tissues, but then developed into different structures. So we can see that ovary and testes, which are our gonads, are homologous structures, and these are going to produce gametes and sex hormones. Then we see clitoris, glands of penis, as well as labia minora and body of penis. These are also homologs. All four of these structures contain erectile tissue that's stimulating during that sexual arousal and orgasm. Some protective structures include labia majora in females and scrotum in males, so they're protecting some of the reproductive structures. And then a couple of glands that we see here are greater vestibular glands and a bulbourethral gland. These are going to secrete mucin for lubrication in both, one is in males, one is in females. Homologs and like where they come from is going to be a little more obvious if we look at this image right here. Let me find it. So if we look at this, and this is towards the end of this chapter, but I feel like when we talk about homologs, we really should be looking at this so that you understand where these homologs are coming from, or what that means actually. So if you're looking at this, this is about a 5 or 6 week old embryo, and you can see both mesonephric and perimesonephric ducts. These are, one is male, one is female duct. And then you can see the genital ridge. Okay, genital ridge is what will develop into the gonads. Now, if you're a male, you're going to stay with a mesonephric duct. That's what's going to develop. And then the genital ridge will become testes. If you're a female, you will keep the perimesonephric duct, and the genital ridges will develop into ovaries. But you can see that both testes and ovaries are originating from the same tissue. And this is what homologs mean, that they originate from the same cell line, but they will develop into something different. And then if we look at what an older embryo would look like, this is about weeks 10 to 12. And then you have the difference at birth. You can see that they're really developed differently. Okay, but I just wanted to show you this, since we're talking about homologs here, that this is basically what that means, that they do come from the same tissues. All right, so the reproductive system does not become fully functional until after puberty, and the general sort of a pathway of functioning of your reproductive system will be that the gonadotropin releasing hormone is going to be released by your hypothalamus. And this is going to stimulate follicle stimulating hormone and luteinizing hormone from the anterior pituitary. Anterior pituitary will then send these two hormones towards the gonads, and the gonads will then start secreting the sex hormones, which in males would be testosterone, and then females estrogen and progesterone is what is going to be important here. Typically, females produce one oocyte roughly about once a month, plus or minus a few days. And then males produce 100 million sperm each day. So quite a difference there in the number of follicles, the number of gametes that we produce gender-wise, okay? So we usually, in females, it's once a month. All right. So let's look at the anatomy of the perineum, that is, the perineum is the diamond-shaped area between the thighs, and so this is what we're looking at here in females and males. Notice that the angle in females of this diamond is wider than in males, and this is because our pelvis has a different shape. This is to accommodate passage of the fetus during childbirth. So males do not have to have such a wide angle there. Make sure that you're able to, to label what is what from this picture. Nothing should be really too difficult about labeling of this image. So that's that. So before we start talking about the specifics of the reproductive system in males and females, what we have to talk about first is heredity. This is important because we're not going to talk about complete heredity yet, but just basics of heredity so that we understand where these gametes are coming from, how the gametes, which are the sex cells, are produced, and kind of why. So we humans have 23 pairs of chromosomes. 22 pairs of these 23 are called autosomes, and we number these chromosomes. We number them in order of their size. That's actually the method that they're using, okay? So we number them 1 through 22, and then the 23rd pair of chromosomes is the sex pair, okay? So the sex chromosomes. The sex chromosomes in females are usually X and X. In males, it would be X and Y. I say usually because we now know of more and more variations in these X and Y combinations, and such. There is a lot of different combinations that there could be, but we're not going to talk about those here. So just be aware that generally X and X is female, X and Y is male, okay? The 22 pairs of autosomes, we call these pairs homologous chromosomes, okay? Or homologous pairs of chromosomes. These are pairs of matching chromosomes. What homologous means, kind of like we've used the term homologous when we looked at homologous structures, okay, and their corresponding structures. So homologous chromosomes mean that they're corresponding chromosomes, they're matching chromosomes. When the first cell of the new organism is created, it comes from an egg cell and a sperm coming together, fusing together. The egg cell has one copy or one set of all the chromosomes, and the sperm cell has another copy of all the chromosomes, okay, one set of chromosomes. So when these two cells come together, then you have a cell that's going to be what we call diploid cell that has two pairs of each chromosome, okay? It's going to be 23 pairs ultimately. One set from mom, one set from dad, okay? So each parent gives one set of chromosomes to the offspring, okay? So the cell that has 23 pairs of chromosomes is known as diploid cell. All of your body cells, except for gametes, are diploid cells. And then these are your somatic cells, your regular body cells. And we use the designation 2N to show a cell that has that is a diploid cell. Cells that have 23 single chromosomes, these are called haploid cells, and these are your gametes. So your egg cells and your sperm cells are the haploid cells, okay? Let me make sure that I have a, I just realized I wasn't looking at the chat. Just to make sure that if anybody has a question here, I can see that. All right. Okay, so hopefully that kind of makes sense. Again, all your body cells have 23 pairs of chromosomes, and then your gametes have 23 single chromosomes. This is important because since the gametes will fuse together, you have to have just one set of chromosomes so that the resulting cell then would have 23 pairs of chromosomes. So what we're gonna look at, some cell division terminology here that's important is shown on this in this table 28.2 here. And I'm actually going to start with that single chromosome right here, okay? So your gametes, your sperm and egg cells would have single chromosomes that would look like this, all right? So that this is a single chromosome. So a single unreplicated chromosome that's found in a cell that is not dividing, okay? That's a single chromosome. A cell that is a diploid cell would have pairs of chromosomes, okay? So right here in this middle section, you see a pair of unreplicated chromosomes. This is, so you would have 23 pairs like this. They are just pairs of chromosomes in a cell that is not dividing, okay? Now, we know that when a cell enters division, when a cell prepares, really not even enters yet, when a cell prepares for division, the cell will need to do certain things to prepare for this division. One of those things is replication of the DNA, okay? So the cell, as it's preparing for division, it will make another copy of all of the DNA that has in it, that it has inside, so that the resulting, each of the new cells after the division has its own complete copy of DNA. So what happens to these chromosomes in a regular cell that is preparing for division is that DNA is copied, okay? So what we get is this, okay? We copy the blue chromosome, we copy the red chromosome. And if we're just stereotyping with the colors, okay, if the red is from mom and the blue is from dad, we create a copy of each, okay? So when you get this right here, this is a pair of replicated homologous chromosomes, okay? And homologous chromosomes are not identical, okay? Just make sure that you know that, and that's why we're using two different colors to show them, okay? The red and blue are not identical. Now, the legs are identical, so, oops, yellow is not a good choice. Okay, so this and this are identical sister chromatids. Okay, so the two red pieces are identical sister chromatids because these, we created a copy of that red one, right? So these are identical. Now the red and blue are non-identical homologous replicated, I guess, chromosomes. Okay, so again, red and blue, non-identical. The two red pieces, identical to each other. The two blue pieces, identical to each other, okay? But the red and blue are not identical because one comes from mom, one comes from dad, and you know, hopefully you know that mom and dad don't have the same genes, right? By the way, I forgot to mention, but these homologous chromosomes, they carry the same genes on them, but they're not identical, okay? So they carry the same genes, meaning the same kind of genes. So for example, it could be eye color, hair color, your height, okay? That's the gene, but the actual code for that gene is not the same, okay? It could be the same, so like both parents could give you blue eye color, for example, but they could be different, okay? So, so again, homologous chromosomes have same genes, same kind of genes, but not necessarily the same code for that gene, okay? Hopefully that makes sense. All right, so let's look at how we create these gametes, okay? How do we get the cells that have only half of the genetic information? And again, this is very important that our gametes are going to carry only one set of chromosomes, because if sperm and egg both were diploid cells, when they come together, then we would have 4 of each chromosome, and that's not good. Okay, having extra chromosomes in humans is not favorable. Okay, that's not a good condition. We know that just having a extra, like a single extra chromosome, can be very significant, can have significant consequences. Sometimes it can be fatal. So there's only a few trisomies that are actually survivable. Most of them are not, okay? For example, trisomy 21, where you have the 321 chromosomes, that's Down syndrome. That one has the least, I guess, out of all the trisomies, has the least significant consequences. So anyway, so that's that. So we need to have just one set of chromosomes in our gametes. We get to the one set of chromosomes by the process of meiosis. It sounds kind of like mitosis, but it is not the same. We're going to see some similarities here, and then we'll compare meiosis to mitosis at the end. So meiosis is also known as the lessening division or reduction division, where we're reducing the number of chromosomes in the cell. The meiosis will start with a diploid cell, and it will end with 4 non-identical haploid cells. The cell that we begin with is going to be either in the ovaries or the testes, so that's the location of the cells. In the ovaries, the cell is known as the oogonium, ultimately like the very first cell, and then in testes it will be called spermatogonium, and we'll look at the processes in detail after we get this general meiosis process out of the way. So during interphase, the cell is preparing for division, the DNA is replicated, and so we get those identical sister chromatids after the replication of DNA. These sister chromatids are held together in not always in the center, but you can see the sister chromatids are held by that yellow piece right there, and that's called centromere. On the centromere is a region which is known as the kinetochore, and the kinetochore is what the spindle fiber will ultimately be attaching to. So that's that. Please know that sister chromatids are not the same as homologous pairs. Okay, sister chromatids are a result of that DNA replication. So we begin with a cell that is a diploid cell. Okay, this diploid cell has replicated all of its DNA and now would have 23 pairs of replicated homologous chromosomes. Okay, in our picture right here, we're just showing 3. If they were to draw all 23, it would be a really, really busy image. Okay, so we're just following 3 of those 23. Since this division is called reduction division, it actually is consisting of 2 separate division processes. One is going to be called meiosis 1, and then that is followed by meiosis 2. And because we have 2 division processes, the phases are also going to have to contain either 1 or 2, so that we know whether we're talking about metaphase 1 or metaphase 2. Okay, so the order of the phases is the same as in mitosis. We're just adding the 1 or 2 after the word. So we're going to begin with the prophase 1. That's the first phase of meiosis, more specifically meiosis 1. In prophase 1, the homologous chromosomes are going to find each other. They're going to, well, first of all, chromosomes are going to condense because you know that when a cell is not going through division, the chromosomes are unwound. So chromosomes wind up together, they condense into that chromosome shape, and homologous chromosomes find each other. They when I say find each other, it means they like literally find each other, and they come close together. When they are close together, they form this structure that we call tetrad. Okay, and tetrad comes from the word four. Okay, tetra means four, so the prefix tetra is four. So if you were to count the single chromosomes in a homologous replicated pair, you would find 4 single chromosomes, okay, in that pair. And where they are, you can see them right here. So 1, 2, 3, 4, okay? So you would have 4 single chromosomes there. So these homologous pairs come together, form this tetrad, and this is something that only happens in meiosis 1. You don't see this happening in mitosis. In mitosis, tetrads don't happen. You still have that DNA there, you have that number of chromosomes, but no tetrads, okay? Homologous chromosomes don't even care about each other in mitosis, but in meiosis they do, and they form this tetrad. When these homologous chromosomes are near each other in this tetrad structure, sometimes what happens is they swap pieces of each other between each other. And that would explain why you see a little bit of blue in that red one and you see a little bit of red in the blue one. What is happening right here is they are exchanging segments between each other. So what we are seeing happening here is in these cells, your mom's and your dad's genes are shuffling, okay? Because those chromosomes came from your parents, right? So the pieces of these chromosomes are exchanged between the homologous chromosomes, and this ensures genetic variation, okay? This is what mixes the genes up a little bit. This is called crossing over, and it only happens in prophase 1. It does not happen in any other phase later on, okay? So this exchange of genetic materials happens here. So that's prophase 1. Prophase 1 is then followed by metaphase 1. Meta means middle, and so what we see is we see these chromosomes, these pairs line up along the middle of the cell. Now, in mitosis, you don't see pairs line up. You see just a single plane of chromosomes. But in metaphase 1, you see 2 rows of chromosomes. So it's 2 layers kind of in there. This is followed by anaphase 1, and anaphase 1 is going to pull the homologous pairs apart. So this is not splitting the sister chromatids yet, it's just pulling the pairs apart. And then anaphase 1 will be followed by telophase 1 and cytokinesis. This is where the nuclear envelope reappears, you see chromosomes unwind for a short period of time, and then the cytoplasm separates, so that's cytokinesis. And you end up with 2 cells. Each of these cells is going to have 23 replicated chromosomes that went through crossing over. Okay, 23 replicated chromosomes. Now, this is not 23 pairs of homologous chromosomes anymore. And since this is not 23 pairs of homologous chromosomes, these cells are already considered haploid cells. Okay. Now, what's going to happen to each of these cells is shown on the next page. So if you have this open in your textbook, this is on page 1108, 1109, and this is going to be followed by meiosis 2. So you know how we said that before every cell divides, it's going to replicate its DNA? Well, every time before it divides, except for when between meiosis 1 and meiosis 2. Okay, so the cells that came from the meiosis 1 will not replicate their DNA anymore. They're just going to jump straight into meiosis 2. Okay, and so here we're following those cells that we got after meiosis 1. And each cell is then going to follow these phases. It's still the same order, the PMAD, prophase, metaphase, anaphase, telophase. Okay, we don't see crossing over here. We don't have tetrads in prophase 1 like we did in meiosis 1. You just see the chromosomes reappear. Then in metaphase 2, these chromosomes are going to line up along the middle of the cell. This is going to create a single line or single layer of chromosomes. And then in anaphase 2, we're going to see those sister chromatids being pulled apart. And then you end up with, after telophase 2 and cytokinesis, you end up with 4 cells, 1, 2, 3, 4. Each of those cells only has 23 single chromosomes. And notice that those single chromosomes are different because remember, this went through crossing over. So those genes have been shuffled. These genes are, or these chromosomes are not identical. And so to summarize this and compare meiosis to mitosis, okay, mitosis gives you 2 identical diploid cells meiosis gives you, and everything except for the word cell is going to be different in this. Doesn't give you 2, but it gives you 4. Doesn't give you identical, it gives you non-identical. Doesn't give you diploid, but haploid. And then the word cells remains, okay? So mitosis gives you 2 identical diploid cells. Meiosis gives you 4 non-identical haploid cells, okay? So that's the major difference between these two. We are now going to look at female reproductive system, and female reproductive system is a little longer than the male. It is a little more difficult because we have to talk about the follicles and the oocytes and such. So let's begin with the female. The main organs of the female reproductive system are ovaries. These are the female gonads. Ovaries will produce oocytes, and they also produce the sex hormones, which are progesterone and estrogen. If we look at the gross anatomy of the female reproductive system, you can see that there are 2 regions, anatomical regions that we can refer to in the female, and those are the vesicouterine pouch. That's this area right here. That's created by the peritoneum, and then so as it lines the uterus, it creates these areas right here. And then we have on the posterior side, there is a rectouterine pouch, and that's this area that's behind the uterus and the rectum. The uterus is an accessory structure. Uterus is designed to house pregnancy, and generally it's positioned like this. So this is an anteverted uterus. Some women have retroverted uterus, which is, instead of being this way as you see it on the picture, it would be positioned like this way, okay? So anyway, so that's the position of the uterus. Now this is a sagittal view, so we're only looking at 1 ovary here and 1 fallopian tube, but ovaries are paired organs. So I've already mentioned the uterus being accessory structure. Some other accessory structures we'll be talking about are the fallopian tubes, the vagina, clitoris, and then we will also be talking about mammary glands when we talk about female reproductive system. So make sure that you are able to label these things, like what is on that image. Again, nothing should be too difficult. One thing that I do want to stress is do not get the uterus and the urinary bladder confused. That is something that students do sometimes switch. I don't think they look similar, but make sure that you know that the uterus is on top of the urinary bladder. And then also the urethra and the vagina. Okay, sometimes students get those two confused. The vagina would be the kind of leading towards the uterus, towards the cervix of the uterus. Urethra is associated with the urinary bladder. Okay, so make sure that you're looking at where the line is coming from. And yeah, that's that about this. Now, this is the anterior superior view of a cadaver. So you're looking from the top inside. And so you can see the urinary bladder there, and then the uterus is looking like it's kind of behind it, but it's leaning on top of the urinary bladder there. The ovaries are, this is a paired organ. They're about the size of an almond, so you can see them right here, one and the other one. And they are held in place by connective tissue. The blood supply for the ovaries is ensured by the ovarian artery and ovarian vein. And the ovaries itself or themselves are divided into cortex and the medulla. So here is an image, a diagram of the ovary. Cortex is the place where we're going to see the development of the follicle. The medulla is generally just going to have the blood vessels and some connective tissue in there, but there are no follicles in the medulla. Okay, so we're going to be talking about some ovarian follicles and then the ovarian cycle. So we cannot talk about oocyte and the follicle development without talking about the ovarian cycle. And then we'll actually kind of merge this together with the uterine cycle as well. That's a thing also. So what is happening to the uterus as the ovarian cycle is happening? So the process of formation of the oocytes in female is known as oogenesis. Oogenesis begins in females before birth. It begins with oogonium, which is a diploid cell. It has 23 pairs of chromosomes, and it begins before birth. The oogonia are going to go through mitosis, and mitosis is a regular cell division. Okay, this is a regular cell division that gives you two identical cells, still diploid cells. So oogonia first will go through mitosis, and they will produce two primary oocytes. Okay, now where we are in relationship to this meiosis thing, in this process, prophase 1, this cell is not the oogonium. This is the primary oocyte. Okay, this is an E. Okay, so the oogonium would be before this process. All right, so oogonium divides, produces two primary oocytes, and then the primary oocytes will enter meiosis if they're stimulated to do so. Where are we? Okay, so all of this is happening before birth. These oogonia, when they migrate to the ovaries, this happens somewhere around week 6 of embryonic development. It's pretty early on in the development. By the time the female fetus is about 5 months, she has produced all her oocytes that she possibly will have in her life, and so they get formed in our body pretty early on in our development. Upon birth, you will have about 1.5 million of the primary oocytes. These primary oocytes will be surrounded by some cells. As you can see on this image right here, there are some cells that are surrounding this oocyte, right? So let's look at this a little closer here and talk about these follicles. So what happens to the primary oocyte is the primary oocyte will get surrounded by a layer of flattened cells, and these flattened cells are called follicular cells. So this is going to be your primordial follicle. So we call this a primordial follicle. All right, that's this right here. We have some of these primordial follicles are then going to change a little bit. The cells that used to be flat like this are going to become more cuboidal cells, and these now are called granulosa cells. And the oocyte itself will form a layer right around it, the cell and the granulosa cells. So it's between those two, and that layer is called zona pellucida. Okay, so as you can see, the zona pellucida right here, it's this layer right here. So we have our primary oocyte, we have the zona pellucida, and then granulosa cells, and this is a primary follicle. Okay. The zona pellucida eventually will harden if that follicle or if that oocyte gets fertilized. So if the sperm penetrates the zona pellucida will harden and will prevent other sperm from coming in. So that's our primary follicle. So we have about 1.5 million of these follicles present at birth. Then what happens between birth and puberty in females is these follicles die. Not all of them, okay, but a bunch of them, most of them actually do die. And so by the time a female reaches puberty, there are several 100,000 of these follicles, about 2 or 3 100,000. That depends on where you're looking. I have seen variations in these numbers, but for me, the average of that comes out of all the sources where, that I've looked at, it's about maybe 300,000 of these follicles. Then what happens when puberty begins, and we've already talked about the hormonal pathway there. So gonadotropin releasing hormone causes your anterior pituitary to release the luteinizing and follicle stimulating hormone, and that is then going to stimulate ovaries to start maturing some of these primary follicles. What happens to these primary follicles then is some of them will respond to the hormonal stimulation. Now, why some of them do respond and some don't, we don't really understand that mechanism. We just know that some of them do respond. It's about 15 to 20 primary follicles respond to that hormonal signal. Then they will mature into a secondary follicle. Secondary follicle still has primary oocyte inside. So what's happening to that oocyte is there are some genetic changes there. What that means is that meiosis 1 continues now, but it hasn't divided yet. And the cells, the granulosa cells around this oocyte multiply. So the layer of these cells becomes larger. Okay, the outermost cells of this follicle are called thecal cells. And the thecal cells are going to release androgen, and this androgen will become estrogen. And this is also going to help with changes that are going on as this ovarian cycle goes on. Okay. As these maturing follicles are getting larger and they mature, they are going to release inhibin and also low levels of estrogen. Both of these are ways to prevent further release of gonadotropin releasing hormone, and this will then stop release of follicle stimulating hormone and luteinizing hormone. This is important because we want to make sure that we are focusing on this round of maturing follicles, and we're getting ultimately just one mature follicle and one oocyte that's released. We don't want to be like continuously stimulating a new group of follicles, okay? Because females, as we said at the beginning, we are supposed to end up with just one mature follicle usually. Some women do mature more than one each month, and that's a genetic condition actually that is passed on from the mother to the daughter. And this could lead to fraternal multiples, which is that runs in family usually. So anyway, so we have now our secondary follicle. As that secondary follicle is growing, some of the secondary follicles will develop into an antral follicle. And our textbook really calls that antral follicle as being something else than a mature follicle. Some places just say antral follicle is the mature follicle or the Graafian follicle. Here they're using it as a different phase there. But basically antral follicle is when you start seeing the antrum in the follicle. And antrum is this space right here, and it's a fluid-filled space that is basically there in that follicle. You can see that the oocyte is not just floating right in the center of the cell, but it is attached to one side of the follicle. And the cells that are surrounding the oocyte are known as the corona radiata. Corona radiata it's that single layer of the cells that are surrounding the oocyte. All of the cells that are surrounding the oocyte, that's the cumulus oophorus, and corona radiata is basically part of that, okay? So that's a little bit of terminology here. Typically, only one of the follicles that were stimulated to start maturing, typically only one will develop into that mature follicle or Graafian follicle or vesicular follicle. You pick your name there. It's one of those, or as I said, some people just call it antral follicle. The mature follicle is the follicle that now contains secondary oocyte. So on the somewhere between here, okay, the meiosis 1 has completed. We got 2 cells after meiosis 1, and one of those cells kept most of the cytoplasm and got half of the genetic material. And the other cell is very small. It doesn't have a lot of cytoplasm at all. It has the other half of the genetic material that will not be used in reproduction. That other cell is known as the polar body, and polar body will die, and it does not get fertilized or anything like that. Sometimes people think that that polar body being fertilized could get you the fraternal twins. No, okay, polar body dies. Polar body does not get fertilized. The way you get fraternal multiples is you get more than one of those mature follicles that release oocytes, okay? The mature follicle is going to generate peak in estrogen, and this peak in estrogen will cause a peak in the luteinizing hormone. The peak in luteinizing hormone will initiate or stimulate ovulation. So this phase of the ovarian cycle where we see the development of the mature follicle, this is called follicular phase because we're focusing on the follicle. The second phase, really, I don't like to call it a phase because it really is just an event, is the ovulation. And again, it's stimulated by the peak in LH, and it generally happens in the middle of the cycle. And when I say middle of the cycle, in our textbook, we're going to be using the average length of cycle of being 28 days. I do want to point out that that is not the same for every woman. So we're just using the average. It just happens to be that average of all the women, the cycle tends to be 28 days, but every woman has a different cycle and should, track and follow what her body is doing, okay? We're just using this for textbook purposes here. So if the cycle is 28 days, ovulation generally happens at the midpoint, which would be day 14. I say generally, this is also different from woman to woman, okay? So some women ovulate early, some women ovulate late, and this can change from one cycle to another. There are things that affect ovulation. There are things that could delay ovulation. And so don't rely on that number 14 being your number for sure, okay? So we're, again, just talking about averages and how it usually happens. Okay, so ovulation involves the release of the oocyte, and as you see here, it is released with the zona pellucida, of course, and then the corona radiata cells also come with it. But the rest of the follicle stays in the ovary, and what happens to the rest of the follicle right here is it develops, it starts developing into corpus luteum. And so this phase after ovulation is going to be called luteal phase. Because we see the presence of this corpus luteum. Corpus luteum begins to release progesterone and estrogen, as well as inhibin. Inhibin, because we now release this oocyte, we don't want to start stimulating other follicles to mature. The progesterone and estrogen are going to help with making the uterine lining thicker. And making the uterine lining thicker means that it's more, it's better for the blastocyst to burrow in should the oocyte get fertilized. It would be blastocyst that is implanting. I know we haven't looked at that. We'll see what the blastocyst is when we talk about development. Okay, but it is a structure that results from fertilization. Okay, so progesterone and estrogen are rising. You would see the rise in thickness of the uterine lining as well. The oocyte, if it's not fertilized within 24 days, it dies. Okay, so that means you have a pretty short window to get that oocyte fertilized. Oocyte only lives for 24 hours after it gets released. Now, sperm can live, I think our textbook uses the 3 to 4 days. I have seen numbers from 2 to 10 days, okay? So again, we're talking about averages. I have seen places that claim that sperm can live up to 10 days. So, but definitely several days, okay? And longer than the oocyte, and that makes fertilization more likely to occur, okay? Kind of makes up for that short lifespan of the oocyte. So if the oocyte does get fertilized, then the structure will be growing, and it would implant somewhere somewhere about 7 days after fertilization or so. If implantation happens, then human chorionic gonadotropin begins to be released by this burrowing blastocyst, and that would be a signal for this corpus luteum to keep doing its job. However, if fertilization and implantation did not happen, the corpus luteum will not get the signal that implantation is happening, and therefore it will stop releasing progesterone and estrogen. It will become corpus albicans, and the drop in hormones from the corpus luteum would cause the release of the uterine lining, okay? The process of shedding the uterine lining is called menstruation. The lining shed is menses. The first menstrual cycle for a girl is called menarche. This happens somewhere around age 11 to 12 on average. We have seen a decline in this age over time for many reasons, but the first menstrual cycle, or the menarche, it's determined by many factors, but just the average tends to be about 11 to 12. But yeah, so that's ovarian cycle. Now, if we look at, and here this summarizes like where you see your primary follicle and secondary follicle and so on, and then primary and secondary oocytes. So you can see that between that antral follicle and mature follicle, somewhere there is where the meiosis 1 completes, and you get that meiosis 2. Okay, so secondary oocyte would be after it enters meiosis 2. Talking about the meiosis and what happens to this follicle, we said that after meiosis 1, we get that secondary oocyte, that's going to be your large cell, and then you get the small cell, that's polar body, that will die. Then the secondary follicle or the secondary oocyte will enter meiosis 2, but it's not going to complete it unless it is fertilized. So it will enter meiosis 2, but it will arrest in metaphase 2. Okay, we'll go through prophase 2, and then the chromosomes will line up in the middle, but that's how you will ovulate this oocyte. Is it's in metaphase 2. Metaphase 2 will not continue unless fertilization happens. Okay, so that's that with the genetics. Now this figure, I don't have it on one picture here, but if you look at page 1115, you can see this all nicely next to each other. You can follow the oocyte, and you can also follow the follicle next to it, so you can see what is when. Okay, so you can see that that meiosis 2. I don't know why they put it up here, because that's kind of too high. Okay, it should be a little lower there. Okay, see right there, and then you can see the actual pictures here. The follicles are rather large. You can actually grab a microscope slide and just look at it with the naked eye, and you would be able to see the follicle. And the oocyte is also a rather large cell. Okay, so that's that. Now, I like this figure right here. This is on page 1117, and this nicely shows you what happens to the hormones, how the follicles are changing, and then at the bottom you also see how the uterine lining is changing. So you can see this right here. Now, we are talking about like the pathway from the primary follicle to the antral follicle. It does take many days. Okay, so notice that from between primary and secondary follicle, it's about 120 days or so. So when we're talking about ovarian cycle, we're kind of condensing this into one cycle, but it actually is a much longer process. So from the time that a primary follicle gets stimulated to getting the antral follicle, it's going to be a much longer time, actually. Okay, so basically we are kind of starting with that antral follicle there, okay, for that one cycle. And right here you can see that growth of the uterine lining here. Notice what your progesterone is doing, right? And you can see your uterine lining is doing the same thing. So progesterone is very important in keeping the uterine lining going. And yeah, so that's our ovarian cycle, and the uterine cycle has, the phases are different. Okay, so they have names as well. And we have the menstrual phase, which is when menstruation is happening. Notice that menstruation is at the beginning of the cycle. So the beginning of the bleeding is day 1 of the new cycle. So make sure that you're aware of that. And this is a cycle, so it repeats. But if I were to ask you, when does menstruation happen? You know, you would say it's before ovulation, right? Even though technically it's after ovulation, but that's from the previous cycle. Okay, if menstruation is day 1 of your new cycle, ovulation is day 14, then the bleeding definitely happens before ovulation. So I just want to kind of point that out, because I will be asking you to classify certain things that happen before ovulation and after ovulation. Okay, but just remember, ovulation is that midpoint usually. So that's that. This figure just summarizes the hormonal flow, the hormonal stimulation of all these events here, which we've already talked about as we talked about that cycle. So this is a nice summary of what's going on. Here we're looking at the parts of the uterus here. The uterus is, and then also the fallopian tube. So let's start with the fallopian tube first. Fallopian tube is also known as the oviduct or a uterine tube. It is supported by the suspensory ligament a little bit, and that one also supports your ovary. The fallopian tube is going to be the structure where the oocyte goes after ovulation. A fallopian tube has 4 different regions here. Infundibulum is the part that leans against the ovary. The infundibulum has structures known as fimbriae. So you can see that right here. They look like little fingers of that infundibulum, okay? And that's basically what their function is to act like little finger-like structures. And these are helping scoop the oocyte in when it gets ovulated. The next section is the ampulla of the fallopian tube, and this is where the fertilization generally happens, okay? So it's in the ampulla of the fallopian tube. Then the next part is the isthmus, that's the green region of that tube, and then the part that goes through the wall of the uterus is called the uterine part. The inside of the uterine tube would look like this, so you can see that it's very branched here, but all these cells are ciliated, and these ciliated cells ensure that there's current created, and it helps roll the oocyte down the fallopian tube towards the uterus. Okay, that's that about the uterine tube, really. There's also a muscularis layer in the fallopian tube, and this muscularis layer, as it contracts, it helps push the oocyte towards the uterus as well. The uterus is a muscular organ, and actually, let me go back to this still. The uterus has several regions. Also, the rounded part of the uterus is the fundus of the uterus, and generally, like during pregnancy, doctors will measure the height of the fundus, and the height of the fundus corresponds to the gestation of the fetus in weeks. So that can tell you whether the growth of the fetus is going on track, okay? Height of the fundus has nothing to do with how much weight the woman gains. I just want to point that out, okay? Height of the fundus grows pretty steadily throughout the pregnancy. So fundus is that top part, then there's body of the uterus, isthmus, and cervix. Yeah, that's that. When we look at the cervix, sorry about that, I have a meeting coming up, so let's try to get through this. The cervix, when you look at the cervix has distinct regions here. We have the internal and external os. These are the narrowed regions of that cervix. And then between this is a cervical canal. And then the cervix is this whole thing, that part of the uterus. Cervix actually changes positions during the cycle. So, and it tends to be a little more opened up as far as the os goes. During the fertile time, and then kind of closes up and changes position during the non-fertile period of time. So some women that track their cycle and chart, they would also be tracking changes in the cervical position and softness and such. During pregnancy, that cervical canal forms mucus, and this mucus would create this mucus plug, and that would prevent bacteria from entering the uterus where the fetus is during pregnancy. But there's normal production of cervical mucus. That's another thing that women chart if they chart their cycle, is how, the thickness of the mucus tells you how fertile you are. But then during pregnancy, it's very thick and creates this plug. Okay. The uterus has several layers. So if we're looking at the layers of the uterus, we have endometrium, which is the internal layer, then myometrium, that's the muscular layer, and then perimetrium is the outermost layer there. The endometrium actually is divided into 2 sublayers, functional and basal layer, and the functional layer is what becomes shed during menstruation. basal layer will become the new functional layer in the next cycle. Okay, so that's kind of how the growth of the endometrium goes. And is that about the uterus? The uterus actually does contract during menstruation. It's like little mini contractions, you know, much weaker than during childbirth, but it's almost like the uterus is practicing for childbirth. Okay. All right. So the next part is next accessory structure is the vagina. This is a fibromuscular tube that serves as a copulatory organ, birth canal, also passageway for menses. And that's that about the vagina. We see non-keratinized stratified squamous epithelium here as far as the lining goes. Let me see. I wanted to, okay. For the external genitalia, just make sure that you are able to label these structures. So labia minora, labia majora should be pretty straightforward here for the external genitalia. Again, this is just really labeling, know what is where. Then, yeah. Really last part of the female reproductive system that we need to talk about are the mammary glands. Mammary glands are a compound exocrine glands that secrete breast milk after the delivery of the baby. The breast milk is fluid that is composed of fats, proteins, lactose, as well as antibodies, and this nourishes and protects the infant. There are suspensory ligaments that support each breast, and these ligaments are connected to pectoralis muscle. There are large lobes that compose this gland, and these are divided into smaller lobules, and each lobule is composed of alveoli, and then these alveoli produce the breast milk. Each lobe is going to be draining into ducts, and there are about 10 to 20 of these main ducts that then come together in the nipple area. You can see right here that right before the nipple, there are these wider areas right there. So the duct becomes a little wider. This is known as the lactiferous sinus. And the function or the purpose of the lactiferous sinuses is that after infant finishes nursing, there will be milk that is left in the ducts and also in the sinuses. So upon the subsequent nursing session, when the infant latches, it gets the milk from the sinuses and from the ducts, and you know, the sinus just allows more milk to be there, and that kind of reminds the baby, okay, this is what I came here for, so I need to start drinking so that letdown can be initiated. And we'll talk about lactation a little more in chapter 29. So yeah, so I'm gonna stop here with the mammary glands. I know there's the last part of the female reproductive system is the female sexual response, but I like to talk about them together with the male sexual response so we can kind of compare and contrast them because there are some similarities, but also quite a bit of difference there since there are different structures that are involved. So, so we'll start with the sexual response later, okay? So any questions? The male reproductive system will be a little shorter than this just because it's a little easier. All right. Any questions? I have maybe like a minute for questions because I have to run into another meeting, so. Okay, nobody's saying anything that they have any questions. So if you have questions, make sure that you email me right away. And I will be, you're welcome. I will be posting a couple of things that you will need to be turning in that they are not posted yet, but they will be coming up. And those are the lab exercise and then the SpongeBob genetics that you will need to be doing. But it's not due it's not due tomorrow or anything. So that is coming up. All right. Well, I'll see you later.